In this topic, we're going to have a look at ATP. So by the end of this topic, you should know what is ATP, what is the structure of ATP, how does ATP store energy, how is ATP synthesized, and what is the chemiosmotic theory. So what is ATP? Well, it's the universal energy currency of all cells. So almost every energy requiring process uses ATP. It's a very small water-soluble molecule, so it's easily transported around the cell. And it stands for adenosine triphosphate. So what is the structure of ATP? Well, we say that ATP is a phosphorylated nucleotide, and it's got three different parts that you need to know. There's adenine, which is a nitrogenous base belonging to the group called purines. Then there's a ribose, which is a sugar molecule with a 5-carbon structure. This acts as the backbone to which the other parts are attached. And then you've got 3 phosphate groups. So in an exam, if you had to describe it, you would have to note down that it's 3 phosphate groups. There are different bonds that you also need to know. There's an ester linkage between the phosphate group and the ribose sugar. And then you've got covalent bonds. These covalent bonds are very important because they're called high energy bonds, which we'll have a look at in a little bit. So if you were to draw out ATP, this looks a little bit confusing, but it's quite easy. You would easily identify adenine. Then you have ribose, which sits between the phosphate group and the nitrogenous group. And collectively, these are called adenosine. If one phosphate group is attached to the adenosine, we call the molecule adenosine monophosphate, mono meaning one, so AMP. If you've got 2 phosphate molecules, we call this ADP, adenosine diphosphate. So why do we use ATP? Well, biological systems transfer the energy in glucose to ATP because unlike glucose, ATP releases its energy instantly in a single reaction. And the hydrolysis of ATP releases a small amount of energy, which is ideal for fueling reactions in the body. So how does ATP store this energy? So notice how ATP's got 3 phosphate groups. And these are key to how ATP stores energy. Each phosphate group is negatively charged, so they repel one another. This makes the covalent bonds that link them rather unstable. It's kind of like a coiled spring. Now these unstable bonds have got a low activation energy. What does this mean? Well, because they've got a low activation energy, they're easily broken. So when ATP is hydrolyzed, for example, water is added and the end phosphate group leaves, energy is going to be released. So we say that water hydrolyzes the bond and the end phosphate group leaves. This is going to give out about 30.5 kilojoules per mole of energy. We're going to have a look at the energy values in a little bit. So notice how the end phosphate group has left, water has hydrolyzed the bond, so you've gone from ATP, adenosine triphosphate, to ADP, adenosine diphosphate. And then PI stands for an inorganic phosphate. So how does ATP store this energy? Here you can see a simplified diagram of the reaction. Usually in living cells, it's only the terminal phosphate that's removed. So if you look at this reversible reaction, you can see that when ATP is hydrolyzed, 30.5 kilojoules per mole of energy is released. ADP and the inorganic phosphate are formed. This reaction can be reversed, so when ADP combines with the inorganic phosphate molecule, you get ATP, and we call that a condensation reaction. So let's have a look at the different energy values. When you have ATP being broken down into ADP, you have 30.5 kilojoules per mole of energy being released. So you have ADP being formed and the inorganic phosphate leaving. ADP can be further broken down into AMP with another 30.5 kilojoules per mole of energy being released. And this can be further broken down into adenosine and only 14.2 kilojoules per mole of energy is released. So notice how in each stage you've got an inorganic phosphate being released, and 30.5 kilojoules per mole for the first two reactions, and then only 14.2 kilojoules per mole. So how much ATP do we use in a day? Well, at rest, an average person uses about 40 kilograms of ATP in 24 hours. But at any one time, we've only got about 50 grams of ATP present in our bodies. And during exercise, ATP can be broken down at the rate of 0.5 kilograms per minute. So this means that a single ATP molecule undergoes over 1,000 cycles of synthesis and hydrolysis each day. So here you can see ADP plus P requires energy, it forms ATP. This is hydrolyzed to form ADP and P. When it's hydrolyzed, energy is released, which can be used for cellular work. So why is ATP called the energy currency? Well, ATP is the universal energy molecule between energy yielding cells, energy yielding reactions in a cell, and energy requiring reactions in a cell. So we say that it's the energy currency of the cell, and it's the trade in ATP rather than making use of a number of different intermediates. So why are energy transfers inefficient? Some energy is converted to thermal energy whenever energy is transferred. So we say that it's lost as thermal energy. Now be careful not to get mixed up between an energy storage molecule and energy currency. Energy currency molecules act as the immediate donor of energy, whilst an energy storage molecule stores the chemical potential energy. So an energy currency molecule would be like ATP. An energy storage molecule would be like glycogen, starch, and fat, and these are your long-term energy storage molecules. Short-term energy storage molecules would be glucose and sucrose. Okay, let's move on to how ATP is synthesized. Now, the synthesis of ATP from ADP involves the addition of a phosphate molecule. We call it a phosphorylation reaction. The enzyme is called ATP synthase. It's also known as ATPase, and this catalyzes the reaction. So ATP synthesis can happen in three ways. You've got phosphorylation, which takes place in the granum of the chloroplast during photosynthesis. So notice on this diagram where the granum is. Then you've got oxidative phosphorylation. Oxidative phosphorylation takes place on the inner mitochondrial membrane of plant and animal cells during the process of electron transport. This involves a series of oxidation reactions that produce sufficient energy to form ATP from ADP and phosphate. And then you've got substrate level phosphorylation, which takes place in the cytoplasm during glycolysis and then inside the mitochondrial matrix during the Krebs cycle. It involves a single reaction where the phosphate group is transferred from a donor molecule directly to ADP. So how is ATP synthesized? We're going to have a look at chemiosmosis. We will be looking at this in a lot more detail in one of the following topics. But if you've already studied it, here's a quick recap. So energy is released during the transfer of electrons along an electron transfer chain. There's going to be a difference in the hydrogen ion concentration on either side of the membrane, and this occurs in both chloroplasts and mitochondria. So if you look at this diagram here, you can see that the hydrogen ion concentration is higher in the thylakoid space or the intermembrane space in the mitochondrion. So what is going to happen? Well, the hydrogen ions can flow down their concentration gradient, but they can only go through a special protein channel, and we call this protein channel a stalked particle. So when they flow through this protein channel, the stalked particle, electrical potential energy is released. And this is used to synthesize ATP. So we call this whole process the chemiosmotic theory of ATP synthesis. So the proteins that transfer the hydrogen ions are called stalked particles. At the bulbous end of the stalked particles, so that bulging end, there's an enzyme called ATP synthase. This causes the phosphorylation reaction so that you get ATP being formed. We're going to discuss this process when we look at oxidative phosphorylation. So let's have a look at a summary. So what is ATP? It's the universal energy currency and it's a water-soluble molecule. The structure of ATP comprises a nitrogenous group called adenine, a sugar phosphate group called ribose, and then 3 phosphate groups. How does ATP store energy? Well, when the covalent bonds between the phosphate groups are broken by hydrolysis, you've got energy being released. Do you remember how much energy was released when the first phosphate group leaves? This is 30.5 kilojoules per mole. And then how is ATP synthesized? So ATP synthesis can happen in 3 ways. These include photophosphorylation in chloroplasts, oxidative phosphorylation in the inner mitochondrial membrane of plant and animal mitochondria, and then you've got substrate level phosphorylation, which takes place in the cytoplasm during glycolysis and the mitochondrial matrix during the Krebs cycle. And then finally, what is the chemiosmotic theory? This is when the hydrogen ion concentration is higher, where? On the intermembrane or in the intermembrane space, so the protons are going to flow down their concentration gradient through a stalked particle, which is a special protein, and the electrical potential energy that they release is used to synthesize ATP. Do you remember what the enzyme involved is called? It's ATP synthase. And that concludes our lesson, the end.